All right, everybody. So we talked about proteins and how they're super important. We talked about amino acids and how they are the building blocks of the protein. So now let's talk about the nucleic acids. All right, we finally get here to deoxyribonucleic acid and ribonucleic acid. All right, it says here in the intro, let's see what color I can use today. I think this light blue is nice. All right. Nucleic acids intro right there with a little arrow. There we go. If proteins are required for the reactions of life to occur and the protein three-dimensional structure is determined by a linear sequence of amino acids, then there has to be a means to make proteins and have information about the order of amino acids for every protein in our cells. Wait, what? Okay, so what that's saying is if proteins are super important, which we talked about last time that they are, and that we talked about how the protein's three-dimensional structure, okay, whether it curls around itself or it makes a flat sheet, remember alpha helix, beta sheet, and then whether it goes past that to curl on some more. Oops, excuse me. I just bumped my table. I'm sure that sounded amazing to you guys. Yeah, okay, so all of this is determined by that linear sequence of amino acids, okay? Linear, again, like we've talked about in math, it means that it forms a line. Remember, we talked about that this was the sequence we're talking about where all of the amino acids will be in a certain order, and that's what determines what proteins are made. That determines the identity of the protein. So if we have all of that, and proteins are so important, then we have to have good information, good instructions in order to create those proteins, right? We have to have that. Because if they're super important, then also the instructions to make them would be super important. So this is where DNA and RNA come into play. Okay? So big star right here. These instructions are coded by the molecule DNA found in the cell nucleus. This hereditary information has to, A, copy itself and be passed on to later generations of cells, and B, have a means to tell how to make proteins. Okay? So every new cell that is made through cell division has to be able to correctly copy these super important instructions. And then once they have the copy, they have to be able to translate that language back out into actually making the protein. So let's talk about these process. Okay? Let's talk about these processes. All right. So here 23 pairs. Remember, a pair is 2, so 46 chromosomes. Okay? 23 pairs, 46 chromosomes. Oops, I kind of wrote over that. Do it better than that. All right, and so these 23 pairs, they code for everything in our body. Every single cell has a copy of the instructions for everything in the body. Isn't that crazy? So, well, it says almost every cell, but pretty much, I mean, by and large, the nuclei of your cells have this information. It's pretty nuts, okay? All right, and the code system that dictates the order in which amino acids are linked together is provided by the DNA, okay? So let's go to that next page. Okay, so let's talk about DNA. Okay, now normally we see DNA as like a spiral. Let me see if I can. Okay, normally we see that, and that's the helix, right? That's the alpha helix shape that we are really familiar with when we start talking about DNA. So what this is right here thing is as if we took that alpha helix and we spread it out as a sheet, okay? So you see here the backbone. 1, 2, 3, 4, 5, 6 little dots over here, and then 1, 2, 3, 4, 5, 6 little dots over here. This is like the sides of the ladder, and then these bonds between are actually the hydrogen bonds between the base pairs kind of form the rungs of a ladder. So when we straighten out this alpha helix, we actually get something that looks more like this. Okay? You can see my sad little train track ladder there. Okay, there it goes. All right, so these hydrogen bonds here, they only form between specific matching pairs. Now in DNA, guanine goes with cytosine. That's what this says right here. This is our first match. And adenine goes with thymine. That's our second match. There are 4 nucleotides, okay? And this right here, hold on. Oh, excuse me. Let's go back up. All right. So, if you see this right here, this part is the sides of the ladder, okay? And this part, the actual nucleotide, is the rung of the ladder, okay? So now let me hop back to my other one. Here we go. Okay. So these rungs only pair up in these specific combinations. Now, you see this 5th one right here. This 5th option, if you can see this, I'm going to underline it. It says replaces thymine in RNA, ribonucleic acid. Okay? Here we have the different numbers of bonds. Ooh, excuse me. Excuse me, excuse me. Okay. So let's start talking about this picture, okay? So that I don't keep repeating myself. How to go from DNA to RNA to protein. Well, DNA looks like the ladder, right? We've got the 2 backbones, and we excuse me, we have the 2 backbones and then the rungs between. Okay, so the first thing we have to do here, when we start with this double-stranded DNA, is we have to, do you see this loop up here? This is your first step. We have replication. So we have an enzyme that helps unzip. You see it working right here. I'm going to color over this particular one as we talk about it. We're going to unzip that DNA right here and start making copies as they go off this way. Okay, so that's replication, making a copy, duplication. Okay, that's up here. A. All right, now we have our 2 copies, right? So then we have copies to pass on to all of our daughter cells, right? When their new cells are made, now we have copies of the information, the instruction manual, if you will, to send with them. Okay, so how do we get that instruction manual to where it can be read and we can actually use the instructions to make the proteins? All right, so that's where RNA comes in. So step 2 here, we have that information and we have to unzip it again. Do you see where it kind of comes apart? There's a gap in the middle here. Okay. we have messenger RNA. This is so small, guys. I'm so sorry. This right here is a little m. Oh, that's very strange. Little m RNA. Okay, messenger RNA. And so what it does is it unzips the DNA at the specific instructions it needs, and then it starts traveling just along the instructions that it needs, and it translates out this information. Now you'll see it here, again, if we're following the arrows, you see how it looks like half of a ladder when it gets to this point? It's got one side and it's got half of a rung, right? So then we need transfer RNA. Okay? We need the ribosome and we need the transfer RNA to come in and help us finish the job. So once the messenger NRA has transcribed, right here, or made a copy of the important information, then we have that copy go into the cytoplasm here, which is the goo in the cell, just cytoplasm in general. And this ribosome goes along and it starts reading this half of the ladder. Okay? And then look here, I'm going to put the star on this side for translation. Ribosome attaches to and moves along messenger RNA to facilitate linking of exact sequence of amino acids to make a unique protein. Transfer RNA brings amino acids to lengthening protein chain. Okay? So if you can picture this as being sort of like a assembly line, okay? The ribosome here is the actual place that calls for the part, okay? So when you have these instructions, again, this is the instructions that look like half of a ladder, so that they're open, they're unzipped, and we can actually read the information in the middle. That ribosome attaches and says, hey, okay, all right, I've got an adenine here, I need a guanine. And then transfer RNA, tRNA, will grab, this is supposed to be little hands, don't laugh at me, will grab a guanine and bring it over to the ribosome, who can then attach it. And then it looks for the next base and it says, okay, so now I need the pair for this base, and I need the pair for this base, and I need the pair for this base. So tRNA is going and getting the stuff off the shelves, as it were, bringing it to the ribosome so that it can be attached. And then here is their output. This long string here says protein. Okay, so as it's going along this ladder, this half of a ladder, it is creating the protein as it goes. Okay, so this says summary here. Let me make sure that there's nothing. Okay, good. Good. All right, so here we have a better picture of the ladder right here, okay? So we have the old strand here that looks like one ladder. Then the next step is when it gets unzipped. Oops, that was a very strange looking arrow. Here we go. When it gets unzipped, then we have new bases added in, so that the next step is to have 2 full copies of the ladder, right? RNA here only looks like half of a ladder. Okay, so we've got the backbone still, and we've got these rungs, put dots around, these rungs that are waiting to be translated with the ribosome, and so on and so forth. Okay, here's another DNA, RNA, protein sort of thing. I don't think that this is any clearer, but if it helps you, please pause the video, study it, whichever. Okay, and we will stop here for today. Because I do want to go over these cellular processes and definitions, but we already are at 15 minutes, so I'm going to let y'all go. This is where we stopped for 4/8/20. All right, and as always, be looking for some questions. All right, I love you all. Have a great and wonderful day.